Everybody, if you could take your seats, we will start session 2. And our chairs for this session is starting with Margaret and her partners in crime in organising this companion animal session. So I'll hand it over to Margaret to introduce our speaker. Thank you.
Thank you very much, Sharon. And it's a pleasure to introduce to you our first keynote speaker for the companion animal session, Gary Whittaker from Cornell University. Gary's an expert in coronaviruses, really, and he's recently published on the differences between the enteric feline coronavirus and the coronavirus isolated from cats that develop feline infectious peritonitis. So we've got a really intriguing title, Gary, Alice in Wonderland and the Red Queen. So we're very much looking forward to what you have to say this afternoon. Thank you for joining us.
Yeah, thank you, Morgan. I do feel I need to explain the title a little bit. So the reason for the title is when I got invited, I was thinking about viral evolution a lot. I've been trying to learn some evolutionary biology over the past year or so. So this is where for those of you who know the Red Queen, this is the model of selection by evolution, and virologists have adopted it to think about this arms race between the virus and the host. So that's the Red Queen is always trying to, the virus is going as fast as it can to stay in the same place, is the concept there. And what I've been trying to do is think about some of those concepts and apply it to what we do, which is a fairly mechanistic approach to thinking about coronavirus spike proteins, and in particular about FIP and feline coronavirus, which is really the most fascinating, I think, coronavirus virus that I've ever come across. So it's really intriguing me. And what we're going to do is think about this in the context of what we study a lot is this now infamous furin cleavage site, which is present in some of these coronaviruses, and think about this, trying to adapt the logic of the Red Queen, and this is why I feel like I'm kind of Alice in Wonderland, like learning a lot of, my eyes are opened about clinical medicine, but also evolutionary biology in the process of doing all this work. So we're kicking off the session on FIP. You'll hear more about this as we go forward, but I think I just want to try and capture everything in one slide. For those of you who don't know FIP, it's been the feline coronavirus has been around a long time, since the 1960s. And one of our famous alums, Gene Holtzworth, identified this in 1963. It was then attributed to a coronavirus, and we had these very intriguing two clinical profiles. We have this benign form, which is known as feline enteric coronavirus, and this highly lethal feline infectious peritonitis form, which in itself can present in two fairly distinct clinical forms, the so-called effusive or wet form and the non-effusive or dry form. So it's considered to be an enteric virus, and these can be fairly prolonged. And unfortunately for cats, in about up to 10% of these cats over their lifetime will develop this FIP form, which is really notably has macrophage tropism. So this triggers the peritonitis. It triggers all the consequences of a macrophage tropic virus. But that in itself is not transmissible. But it's the transmissible form is the FECV, and there can be relatively long-term persistent shedding in different numbers of cats over the life of the cat. So a complicated disease profile, complicated even more by the possibility of antibody-dependent enhancement for the FIP form because of the macrophage involvement. So that's the challenge, and the challenge as a experimental virologist is that really it's not possible to isolate these viruses in at least the clinical strains in cell culture. So this has really held back advances in the field for this reason. Because of that, there's no vaccine, there's very limited molecular testing, and no approved antivirals. You'll hear a lot more and I know in the next talk about there are antivirals available now, but at least in the United States they're not approved. And so I'm not going to really touch on that at all in this presentation. So to summarize everything, then we have these two bioforms, the FECV, the benign, and the lethal form. But really what I want to emphasize right here is we have these two, there's also two serotypes, which are called FCoV1 and FCoV2. And the note about the not possible to isolate really applies to FCoV1, which is by far the most prevalent clinical strain that's circulating in the cat population. So this is where we are in terms of phylogeny. We have, it's an alpha, I should point out, it's an alpha coronavirus. So SARS-CoV-2, for instance, will be down here, along with all these, bat and bat-related and animal viruses and beta coronaviruses. So these have been very well studied, certainly over the past few years. We have the Delta coronaviruses in the middle. You heard about those this morning, and the gamma coronaviruses too. And then we have this big cluster of alpha coronaviruses, including some human viruses and a lot of animal viruses, and then alpha coronavirus 1 is a single species, according to the taxonomists, which includes all of these quite wide range of different feline, canine, and porcine coronaviruses. But really, I think the idea of a serotype is too simplistic, and I tend to refer to these personally as different genotypes. So we have the type 1 at the top here, which is a distinct genotype if you look at the branching of the tree to this virus here. Because there's really very little similarity in spike, it's not just a small change. There's a big change in spike in the process of discriminating between those two viruses. And just to re-emphasize, this is the virus we're going to concentrate on for the rest of the time. One of the big differences then, just to come back to this furin cleavage side, that's where what got me intrigued many years ago is because we have this very obvious difference between the furin cleavage site. This is this boundary between the S1 and the S2 domain. It's this activation site for a protease, so we look at arginine residues, arginine serine pairs in particular. And if you look at the beta coronaviruses, the gamma coronaviruses, these are prevalent or at least quite common depending on the virus, but they're essentially missing from all alpha coronaviruses. If you do an alignment, there's a big deletion or a big missing piece of the spike protein right there. So the fact that we have a furin cleavage site embedded in this alpha coronavirus lineage is very unusual and very intriguing. And that's kind of what got me really interested way back in terms of following all of this up. I'll just note at the bottom here, when I come to the biochemistry of the cleavage event itself, I may refer to these P numbers. That just is the protease will come in here between P1 and P1 prime. So this will be an arginine, and then we give it a count in different directions in terms of non-primed or prime. So just as a little reference point to the nomenclature as I move through the slides. So that first slide was the last slide was done several years ago before SARS-CoV-2 came along, and then as I'm sure most of you know by now, we have this insert, this 4 amino acid insert sitting right in that junction. So we don't have an RS sequence here with SARS-CoV-1. We have a RRA-RS sequence here. So that's the furin cleavage side of SARS-CoV-2, which is really allowed this virus to be transmissible compared to SARS-CoV-1. And when we put that into structural perspective, this is a nice little summary of the way I think about what's going on here. We have a very dynamic spike protein. These things are not locked into position by any means. They're very dynamic. And we have I think essentially a big range of open and closed conformations. This is a trimer. What's shown here is that one receptor binding domain, or RBD, in up conformation. These can be 1, 2, or 3, depending on the situation. And really, the furin cleavage site, which gets inserted, really relaxes the spike protein, allows much more up conformations to occur, and really converts a closed conformation spike, which would be more typical of SARS-CoV-1, into an open conformation spike, more typical of SARS-CoV-2, allowing then the RBDs to go up, and it allows much more efficient receptor engagement, and that really drives the transmission, and the virus is sitting in this transmission window. But it's a little dangerous for it to do that, because as the RBDs go up, the spike is actually pretty unstable, and the S1 can then also fall off. So it's a bit of a risky endeavor for the viruses to do that. It needs the open conformations to transmit, but it also has this risk of actually falling apart in the process of doing that. So it really has to sort of sit in this window. There's a window of opportunity that SARS-CoV-2 has really taken advantage of and is continuing to sort of work through as the pandemic proceeds. And just to make a note, if everybody's read some of my articles, I've got linked into this whole lab leak theory. So I want to point out that this is it's not the smoking gun. It's way more complicated than that, and really it's really part of natural selection. And what we're trying to do is trying to understand how this selection is proceeding and really build that into a model of the origins of SARS-CoV-2. So this is an article that I kind of had to rewrite and reissue in a way just actually a couple of days ago. You can't read this very well, but just a couple of days ago this got updated in the Lancet Microbe. So in terms of this stability, then it's very, very clear now that SARS-CoV-2 will put this furin cleavage site in there as part of natural selection, but it will also take it away very readily. And this is what happened with probably 3 years ago now, when people like me started to grow SARS-CoV-2 in vero cells. Then we rapidly realized that it was deleting that region, and a lot of people figured this out, and either through little deletions as shown here, or point mutations, and it can happen very quickly. This is a passage 3 in a vero E6. We've had the same. These point mutants will come up in one pass in a vero temporis 2 cell. So the virus really, and this will make the spike uncleaved and adapt it to what's likely to be a cathepsin-dependent pathway instead of a temporis 2 dependent pathway. So this happens very, very rapidly in cell culture. So this is the concept that I want to work through in terms of what might be going on in a cat with feline coronavirus. So trying to build in some of these concepts of up and down conformations and adaptation to cleavage sites as we go through different scenarios for FIP. Okay, so this is where we go down the rabbit hole. We're back to Alice in Wonderland, and I'm going to go back 10 years or more now, and almost like 14 years to the person who sent me down the rabbit hole, which was Ed Dubovy. Some of you may know Ed Dubovy. He was the former director of virology at the Cornell diagnostic lab, and he came to me and said, I've got a ragdoll breeder with an FIP problem. I don't know what to tell them. The tests aren't very informative. The serology is not very informative. What do I do? So he gave me a bunch of samples, these fecal samples that had come in, and I did some sequencing, and we spotted this motif right at the junction of S1, S2, and this gave me for sure a bit of a surprise, because back in 2009, everybody was thinking more about bird flu. The last time that H5N1 was going through the bird and chicken population, and this is where we get these so-called polybasic sites are very distinctly localized to H5N1, and it's the same thing. It really switches tropism very readily in H5N1 to make a highly pathogenic avian influenza. So my first thought was, we've got some equivalent thing going on here. We've got some highly pathogenic FCoV going through this breeder, and we've got a real problem here. But thankfully, quite rapidly, I realized that wasn't the case. I did some research, got some samples from another lab, and realized that this was fairly normal. This was all in the context of FCoV. This so-called polybasic site was very normal. And really, all the fecal samples we were looking at had that sequence, and that was subsequently published. And this is now going back 10 years, so I won't go through a lot of the details. But we took a range of FECVs and saw a very conserved motif of RRS. This pairing of arginines and serines is very distinctive. And then when we went to the FIP situation, these were all pathology samples from FIP cats. And we can see that it's like that situation with the vero cells. Scattered in there, within that motif, there are point mutants, threonines, methionines, glycines, various, but it was almost every cat was different. So it was never a consistent pattern. We've got what looked like a loss of function, but a very consistent loss of function and disruption of this loop. Right, what turns out to be a structural loop in that region. So that was that's 10 years ago. We made what we thought was a breakthrough, and it's been frustratingly hard to kind of move on from that initial finding, because it's a really complicated situation. It turns out way more complicated than we ever realized. But it's some examples of this. We've gone through the published work is all summarized here. So we've gone through different scenarios. This is a dry cat with neurological signs, and we get this kind of mutation. We get an arginine methionine, but only in the central nervous system, along with the IHC. This was a more consistent arginine to serine in a variety of different tissues. So it's almost like every time we look at a cat, we get a slightly different situation. And the one I'm going to focus on here is what appeared to be an outbreak. And it is known that in the context of breeders and shelters, there do appear to be what are outbreaks, but there's not a classic transmissible outbreak. And this was one of those situations where we had it was a house, an owner with at least 7 cats in a situation outside of Whittaker, and the house caught fire, and there was a big stress situation. I think this is, if anything, is going to trigger FIP, whatever stress means, and cats get easily stressed. But some stress situation is what will trigger the FIP. And what we think is the situation here is we have a circulating FCoV, and then we have a big stress situation for the cat. They have to flee the fire. They get taken to the shelter, and then right away we get these pathogenic variants get selected. But each cat is different, and within individual cats, then it's not completely different across tissues, but there's a collection or a constellation of different mutants that get selected in different tissues, but all with the same pattern of somewhere in that cat there's a sequence which is has a down-regulated furin cleavage site. That's been the consistent message over the past 10 years or so. But it's clearly, I'm focusing on the furin cleavage site. It's not entirely that situation, and so we have to keep in mind other mutations in spike. And this is an early example of a cat that we sampled. We're working a lot more on getting longitudinal samples, but this is a very early 2-year timeline. We have one of the original cats from 2009 was sampled, and 2 years later it got FIP. So we did a full spike sequence, and here's our furin cleavage site mutation here, R to L. We have an M to L mutation. This is also part of the picture. We have the M1058L residue. There's another cleavage site I didn't mention, it's S2 prime. That does change, but it doesn't always change, and it didn't change in this cat. But overall, we have 60 mutations over that 2-year timeline, including a big chunk of change up here at the N-terminal domain, and which looks to me like recombination. We have to, these viruses recombine as part of their lifestyle to recombine. So we have to think about recombination, we have to think about mutation, and we have to think about is this a reinfection, or is it the same virus that is now present in that cat 2 years later? And what I'm thinking, I used to think that it was more reinfection, but now we're changing our minds on that a little bit, and in part because we've been very fortunate over the past year to bring in Raina Plowright to our disease ecology group at Cornell, who does a lot of work on bat coronaviruses. And when Raina was giving her job interview, she pulled up this slide, which is from one of her studies, and thinking about how bats will maintain coronaviruses. And it's, this is a typical SIR model of a recovery. This was, I think at the time, the prevalent model for feline coronaviruses. The cats were recovering and they're getting reinfected. But I started to think maybe the cats are more like this model. Maybe it's what Raina was calling an SILI model, or a silly model for there's an initial infection, there's some persistence or latency, she was calling it, and then in the case of the bats, we get stress and rebound and shedding of the virus from the bat caves, and this continues. And so this is, we've in a way switched over to this kind of model to think about how cats might work. And if I animate this through, there's that mapping back to that situation of cat number 234. What would have happened here is that there was an initial infection, some persistence, and then the macrophage tropic variant arrive and the cat dies, and that's kind of the end of the story. But now we're certainly in a whole different ball game in terms of how we're managing FIP with all the antiviral drugs coming along. So we have to think more about really what's going on in terms of within the cat. What are these viruses sitting in reservoirs? And then when you treat the animal, we've got more of these situations now where the cat could be treated, and recover, but is it really recovered? Is it really cured, or is it really just in remission? And is that virus just pushed back to its reservoir? So we'll find out in the years to come in terms of really what's going on and how to manage these situations. So with that being said, as I say, I've been thinking more about viral evolution, at least talking to people who do know about viral evolution, including one of the very fortunate collaborators we've picked up is Sergey Pond down at Temple University, and one of his students, Jordan, who's now moved to Cornell for a postdoc, so I'm very excited about that. And what Jordan went through his thesis, like picking interesting coronaviruses, and chapter 1 of his thesis he just defended last week was canine coronavirus, then chapter 2 was feline, and then chapter 3 is equine. So we're thinking like really mapping all this evolutionary biology into relatively limited sequences in terms of trying to adapt all this work from SARS-CoV-2 where there's millions of sequences down to maybe dozens of sequences. But the Pond Lab have these really good tools to be able to statistically work through samples even in the absence of large numbers of sequences. So we've been very fortunate to be able to work with Jordan and Sergey. And so this is the outcome of this paper was just published. This is the outcome. And what Jordan did was to take all the genome sequences of FIPs and FECVs, map them onto the structure and look at the sites of selection. And we heard about this morning in terms of purifying selection and so forth. So this is the outcome of all that work. And what we have is two residues. The blue residues are really the sites that really evolved separately between the two viruses. So the ones here, that 1058 residue I sort of alluded to, but also within the furin cleavage site. But a lot of all these purple is a lot of adaptive mutations right in that furin cleavage site. So this was very good independent verification of the model that we'd been thinking through more biochemically in terms of how really what's going on in terms of how these viruses evolving. And that was Jordan's summary, and I've just modified that a little bit to say, just to reiterate that we've got a very conserved pattern of these arginines and serines in FECV, especially in these residues that are in that turquoise box. These are the key furin cleavage site residues, which are really very conserved in FECV and change a lot with FIPV. So Jordan picked out this so-called P4 residue that's the turquoise residue right here. That was the site that was under most pressure to evolve and in between the two viruses. So that was, again, very, very nice to see that independent verification of the models that we were working on. I want to spend just one slide or two just on what really is a furin cleavage site. As I said, it came out of the bird flu world and virology in terms of this polybasic site. So I want to stress it's much more than that. It's a very complicated pattern. We have, and this is some computational prediction based on peptides that we turn to a lot in terms of trying to understand what's going on here. So we have the purple and green residues will be the furin binding pocket. But we also have a lot of residues outside of that furin binding pocket that can make a difference even at a peptide level. And so it's even the core that we were focused on needs to be expanded out. So we need to be thinking about at least a 15 to 20 amino acid chunk of sequence, which is really controlling how, at a fairly refined way, how furin will recognize that sequence and cleave it. And it's not just on-off switch. There's a lot of sort of inherent regulation or flexibility within that switch. Fun fact: this is called P2, and this is an anime character. P2 is an anime character, and if you run the program and you get a positive score, then the computer meows. So I don't think this was at all intended by the author, but it really fits well with the feline coronavirus analogies. And then this is just a quick note to say, what we've done is, as an experimentalist, I've gone through this is a sort of a subset of all the papers that have been published over the years. And what else, I'll just quickly state here that if you look at the red mutations here are the ones that you can go down and including our own samples now, very strongly predictive of an FIP mutation. So that's great, but the issue is that there's also these, what I flagged as orange mutations. So there's always residues in these residues outside of that binding pocket, which appear in the FIPVs would also appear in the FECVs. So these are what I'm calling transitional mutations. This is the next challenge, to really try and understand what's going on there. But there's certainly a pattern, and prolines come up a lot, histidines come up a lot, leucines to some extent. So there's a lot of selectivity in making that switch. It's not, like I say, it's not just on-off. It's selecting some transitional sequences which amplify the change, if you like, is the best way to view it. So that's the challenge right now, is to try and understand how these transitional mutations that really feed into the whole system to really drive this process forward, and in a relatively complex way. And to do that, we need to, peptides alone are not enough. Peptides are great, but we need to do structural modeling. And just some examples here. This is we're starting to build these models now, and this will be a nice sort of fairly conventional furin cleavage site in SARS-CoV-2. You can see a nice exposed loop with a nice spacing of P1 and P4 residues. So that's a good cleavage site. And then UU4 is our prototype FIPV, where we have a cryo-EM structure, so we can build the mass models. And you can see this is really scrunched in and pulled in, and the P1, P4 residues are kind of pulled chemically there. Then there's a mutation here. This will be the P1 glycine, but also structurally they're kind of retracted and pulled away from accessibility to the protease is really and then again, moving forward, we can build the models that we need to dock furin. And I'll just very briefly mention mouse hepatitis virus. This is our prototype, and for those of you who know MHV, there's a lot of very detailed studies on MHV with different furin cleavage sites with different pathogenic properties. And what we're in combination with our collaborator down at Cornell, Harold Weinstein, he's been able to go in and really explain in really good detail why certain MHVs get cleaved and why other ones don't get cleaved. And it's really very down to very precise changes in the furin cleavage site and how those residues dock into the enzyme of furin. So again, for the future, this is just in a way a teaser for what's coming with our work on FIP. And what we're doing right now is we've picked out 6 different versions of cats with feline coronavirus or FIP, and we've got sequences of these, and we're trying to figure out, look mechanistically as to really, can all those structural models explain the disease in terms of understanding how these are all working. How am I doing for time, by the way? I don't want to go too long. So I'll go through this fairly quickly. 576 is one of our most fascinating cats. It's this cat that it was a 5-year, one of our first longitudinal samples. It was never an FIP cat, but the owner was suspicious and did a lot of sampling. But the clinicians and the pathologists were saying, absolutely not, this is not an FIP cat. But it was definitely positive in certain tissues here. Back in 2017, things went quiet for a period of time, and then the cat died, and we got the body. And what I'll note is that we've got virus in the heart. So this was a positive sample in the heart, and this is, there's clearly, I think there's some, as with SARS-CoV-2, to be honest, there's clearly some link to heart problems. This is just the sequencing is not easy. We have to do this hybrid capture enrichment technology, and the data's, I'll just quickly go through this because of time. But you can see that the feces is a fairly normal sequence, and the nasal biopsy in 2017 had a mutation, and the heart had 2 sequences. So this is actually evidence, I think, of evolution within the heart, even in this sort of so-called reservoir situation where obviously not causing a problem, no myocarditis, nothing obvious, but clearly the virus is present in the heart. And then probably more intriguingly, the conjunctiva had an insertion. So this is pushing the loop out into space. So this is the opposite to the UU4 sequence. We've got a cleavage site which is being pushed out into space. It is more accessible and probably driving that open conformation more than the closed conformation. So this is the endpoint. I won't go through this network analysis to trying to look at the evolution of this, but we have these nodes that seem to drive evolution, and I want to get to the model. So this is the original models here. What we've come up with now is this new model of which is situated here, where we have these 2 loops. So we have FECV sitting in the GI tract, and then we have a loop which could end up in FIPV, but doesn't have to end up in FIPV in the down conformation or the closed conformation, and a separate loop out in this situation. So this is being mapped then into the alpha coronavirus spike. So we have to think about how these domains are being organized, what domain is going up and what domain is going down with an alpha coronavirus versus a beta coronavirus. So it's going to take a little time, but and we need more structures, but this is all work in progress. So here's the red queen. We've got the red queen heading this way, but then we've also got this short-sighted evolution. So that's the concept with FIP, it's short-sighted evolution. With certainly one outcome of FIPV in a macrophage, but also other outcomes in other tissues. You may have noted I've not mentioned immune surveillance at all. So this has to be built into the pictures in terms of really how is the immune surveillance going on. And we have very little idea of what's going on there. I'll skip through, because I know I'm out of time, and I'll come to this, I guess this last slide. So this is what we're trying to do then. We're trying to map the dynamics of what we know is going on with SARS-CoV-2 to a feline coronavirus. We've picked out 6 or 7 viruses to work on, to try and work through the structural biology of this and get it into a wet lab version of what's going on with FIP. And there's lots to do. I think it's fascinating in terms of how to understand this and really think about how this virus could be adapting so readily in the context of a live animal to cause these various pathogenic outcomes is really going to keep me busy for a very long time. A lot of people to thank here. And this, I've come back from Switzerland. This is my former lab, my former lab, all nice in their own labs now. And this is taken in Montreux, Switzerland, the Nidovirus conference last week, where there was a lot of really good studies on coronaviruses, but not many on FIP. And I think I'm convinced a lot of people there that FIP is very cool. And I know you all do too, so I'll end there and take some questions.
Well, thank you so much, Gary. That was fabulous. I think, given the interest of time, we've got time for one quick question. Cyril?
Thank you for this really fascinating talk. I'm just curious in terms of viral evolutionary understanding. Is this furin cleavage site, you said, it's not just a polybasic site, but this phenomenon, I mean, is seen in other viruses, right? With, for instance, influenza viruses. Polybasic site acquisition allows a influenza virus to go from low pathogenic to a highly pathogenic virus, also going from being relegated just to respiratory to going systemic. Mm-hmm. We see the same thing with virulent Newcastle disease. So is it structure function? Can that be used to potentially look at the emergence of new viruses that acquire these polybasic sites? Is it that simple? I know in your talk you said it's a lot more complicated than that. And there's other receptors, but just curious.
Yeah, the Newcastle and influenza situations are much more simple. It's just there's a much more chunk of material that goes in and out. H9 viruses are more subtle. H9 viruses are more like coronaviruses, but so it can be used. We've just got to think very carefully. It's with the coronavirus, we've got bat viruses generally without furin cleavage sites. That I think is the spillover event, and we need to think about cows. We need to think about horses. A lot of viruses have these furin cleavage sites in a lot of animals. Some do, some don't. But if we can really understand what's going on there, it can really be used as a predictive tool to kind of look at high risk outbreaks versus low risk outbreaks, and hopefully make some risk assessment tool, kind of build it as we move forward.
Thank you so much, Gary. So our next speaker this afternoon is Emi Barker from Bristol. Emi's a referral clinician at Langford, and she has a real success story to tell us about the shifts in outlook for cats diagnosed with FIP. So we look forward to hearing the latest from Emi.
Thanks, Margaret. Can you tell I'm a cat vet? So I really enjoy kind of being invited to speak here. I know some of you guys in the audience, and it's great to kind of meet quite a few other people as well. And one of the reasons why it's so great to be able to come to talk to you is what I've been able to talk about FIP over the years has changed significantly, and that's what's so lovely about it. I have a few things on there that I think I'd like to tell you about in terms of who I work for, what I've done in the past, but I'll move on to the other stuff. There's lots of people I've worked with. So up until a few years ago, most of the talks that I gave to practitioners, to researchers on feline coronavirus and FIP tended to stop on about this slide. They'd be doing their diagnostics to try and work out whether or not there was enough justification to euthanize the cat that they had in front of them. These were usually young cats, somewhere between 6 and 3 years of age, cute little balls of fluff that over a matter of days or sometimes weeks deteriorated to the point where they were moribund and needed euthanasia. Most of these owners are utterly devastated by that, and I suspect there may be people in the audience that have been through that trauma of having cats like that. Okay. And one of the real difficulties is trying to prove that without seriously putting the welfare of the cats that were in front of us under a lot of stress. That has changed. That's changed quite dramatically. It was changing before the pandemic was happening. I remember as an undergraduate student, the writing on the wall saying that, there is going to be a pandemic, there's going to be an RNA virus that escapes. And Big Pharma were really switched on to that. They knew that something like this was going to happen. They were looking at various different drugs that were going to be able to be used under these circumstances. And over the years, a number of different drugs have been looked at with treatment of FIP in mind, and these are kind of written on here at the moment. Quite a few of these studies were in vitro work, particularly when it comes down to drugs like cyclosporin, itraconazole. But then more recently, particularly the work that's been done out in the States, we started to hear about GS 524, which I'll call GS, because otherwise it's just a lot of numbers after there. And then more recently, we've also had GC376 as well, which is a protease inhibitor. Now, some of the things you can see on here, really high safety margins, very specific in terms of their targeting these RNA viruses. Some of them less so, more of the narrow safety margins, but they're definitely out there in terms of the drugs that we've been using. So what started to happen around 2018 is that these first papers were published coming out of the US, talking about the use of GS for the management and treatment of FIP. And it made the disease go from being almost 100% fatal to the majority of these cats surviving a disease, something that should have killed them within a matter of weeks, and these cats were living. Very shortly after that, products were starting to become onto the market, marketed at clients for the treatment of cats with FIP. Vets were completely cut out of the story at this point in terms of access to them, because they knew that if they sourced these medications, they were at risk of losing their license and ability to practice. Most owners were terrified of telling their vets because they thought they were going to be reported. So there were quite a few vets on the side that were quietly supporting owners, but they didn't usually put any notes down for those things. And there's a huge market with online information, social media support groups enabling people to treat their cats. And actually what was quite interesting around this time is that this almost ended up in a self-policing situation where any bad players in the market that were selling medications that were no longer effective actually got called out on it. They ended up with a really bad reputation, and people wouldn't buy the medication from them. So it was very much a self-policing situation. And that's the time when I first started seeing a lot about this stuff happening, and we were quietly speaking to vets on the side going, if you diagnose a cat with FIP and you think it should be dead, and you see it a few months later looking amazing, you maybe didn't get it wrong. Maybe that there are these medications that these owners are sourcing for these cats. But it was very, very difficult at this point. As vets, we knew that there were drugs that we could use, but didn't have access to them. Then COVID-19 happened, and Gilead was able to then bring remdesivir to market as Veklury. And once it was available as a human product, we started niggling away at the company, and eventually we managed to get hold of Veklury as a product to be able to use with treatment for cats. And the main reason is that the market they thought that they were going to have for humans wasn't there. It wasn't so effective. Probably the mechanisms between the virus and the host are going to be different, and we do believe that. So we started being able to have access to Veklury. A little while later, starting off in Australia and then more recently in the UK and other areas in Europe, we started having access to a generic form of remdesivir by injection by a pharmaceutical company. And then more recently, we started getting hold of GS as well. We've followed over 300 cats through contacts with other vets in the UK, through support networks that we have in the UK, supporting vets use these medications. And actually what we're seeing from a survival point of view is that we are now seeing in excess of 85% of cats living. They have 3 months worth of treatment, and then they're still alive months and months later. We don't have extended survival times yet, because these cats, we're kind of just coming to the end of treatment for some of these cats. Some of these cats had finished their treatment off towards the end of 2021. When we look at the survival curves from them, we can't get brilliant data yet because these cats are still alive. And that's what's so amazing about the situation that we have. But we have learned a few things about it, that we know that we've got to get the diagnosis right. We need to know what form it is, whether it's affecting the brain, whether it's affecting the eyes, or just the vasculature, because that will influence the drug dosages that we use. So there's still a way to go in terms of diagnostics to try and work out what the optimal doses are going to be. But we know that when we've looked at our cats and gated them for the different manifestations of disease, we know that actually the survival times are the same. Whether we look at a cat that's got neurological disease and we're giving them higher doses, actually the survival time in those cats is the same as cats with effusive disease that are given lower doses. So it does seem to be, if we get the dose right, we're getting the survival times the same. We now actually have 2 manufacturers in the UK that are producing this product. And over the next 12 months or so, we expect to see other antivirals coming onto the market, both generic molnupiravir and its metabolite, and also GC376 as well. Assuming the company that's trying to kind of work on it, I know they're trying to work on it in America, and it'll be great if the Americans can get hold of legal antiviral drugs for the treatment of cats with FIP, because at the moment, what's happening is a lot of owners or cat guardians are importing these medications individually, out with quite a lot of oversight, and at their own risk as well. So how I've managed these cats and how there has been such a change to it is that we've now gone from that, how do I diagnose and be much more comfortable at euthanizing cats that I have in front of me, to at what point do I start reaching for the medication that I have on the shelf? Now, it would be really, really great if we can get down to the bottom and be super confident with our diagnosis, because we know from a medication safety point of view, so that we know that we're treating the right cats, that we're not using these medications indiscriminately, we really want to get right down to the bottom, kind of confirmed disease process. But the issue that's facing our profession at the moment is primarily financially related. With the issues with cost of living, people are not able to spend so much money on doing some of those diagnostics. We've also had vets that are trying to cut corners, and they see a young cat that's got a fever or a young cat that's got abdominal fluid, and they're thinking, oh, it must be FIP, I've read about it recently. I'll treat it with this medication. And actually, we have to be really careful to try and get that balance correct. And I'm not sure we've got it there yet, but certainly my confidence in managing these cats is I tend to start wanting to start treating them slightly earlier. I'll often start collecting the samples, but I'll start treating them whilst those samples are being processed, once I've made sure that they've not got septic bacterial infections in them, or another disease process like a lymphoma, for example. Certainly looking at the data that we have, if we delay treatment in these cats, whilst we wait for results to come back from a laboratory that might take 7 to 10 days, we are losing the battle in trying to fight the disease process in these cats. Median survival time for a cat that has effusive abdominal disease is about 10 to 14 days. And if we try and factor that into our diagnostics and sourcing of medication, then we run the real risk that we will have spent thousands of pounds worth of owners' money and telling them with the carrot going, we have this medication that can cure your cat, with the medication arriving as the cat is euthanized, which is a sad process when that happens. But there are other issues with it. As we said before, we now have these not so legal access to antivirals. And actually what I found quite interesting in the UK is that despite there being legal access to medications, I am aware that there are a small number of people who are sourcing illegal versions of these medications. They're still being supported by online groups, but they're still able to get hold of them. Now, part of that sourcing of the medication is going to be because they cannot afford the drugs that are available on the market at the moment, despite the fact that a lot of vets are actually not charging as much or not marking up these medications as much as they would mark up, say, antimicrobials, other antimicrobials such as amoxicillin and things like that. But they're still trying to get hold of them. We also have owners for whom they may not have trust in their vet going, I don't think this is FIP, I think this is something else, I would like to do some more diagnostics. They are reaching out to people online who might not have the full picture, who are supporting these owners in their belief and then helping them have access to illegal antivirals. We know that this is happening as well. As we said, the market is relatively self-policing, but it still costs thousands of pounds for worth of these medications. It just tends to skim just a little bit under what it would normally legally cost for these medications. In terms of access to these drugs and the support that we have behind their use, both legal and illegal, there are certainly increasing numbers of papers that describe the use of the products that have been sourced by clients. It has been really, really helpful from a clinician point of view that we've been able to see how good these drugs have been working. But it ends up being in a really difficult position. A lot of these studies are retrospective, looking at what these cats have been doing over time. So there is a lot of limitation to the data that we have out there. We know that some of the drugs that have been used in these studies might not have been exactly what they've said on the tin. When sometimes people have looked at the types of medications in the product, they may be a subtly different nucleoside analog. In the earlier stages when some people thought they were using GS, they were actually using molnupiravir. Had the same effect in the cats in that they were surviving the FIP, but they were using a different drug and not necessarily being aware of it to start off with. We also know that the quantities, the concentrations of these antivirals in the medication are higher than that which is actually written on the side of the packet, if they've actually got anything written on the side of the vial that arrives to them, which is not a given. And you might think that's a bit odd. Why are these drug companies giving out more drug than they're actually selling to the client? And actually it means that these clients are giving a higher dose of medication, of antivirals, than they think they're giving. And it means that for these cats with FIP, they tend to be doing a bit better. They're more likely to survive the disease process that they have. And that company is going to get a better reputation with the online communities for managing them. Okay. So it might seem a little bit cynical, but that seems to be what's happening with these medications as well. It has had a knock-on effect, particularly in the small animal community, in that it has really undermined some of the vet-owner relationships that we have, particularly where vets really want to get the diagnosis really solidified, because we want to be really careful with the antivirals that we're using, but where we have owners that are trying to preserve the funds that they have for the thousands of pounds worth of treatment that is required to treat their cats. And we also know that there's the problem with oversight as well. We do have vets in the UK where if we tell an owner that we're not comfortable about a diagnosis, that we don't want to dispense an antiviral medication, we're also in a situation now where owners can source this medication themselves. And then they will do that without our oversight. So it used to be we used to think about the use of antimicrobials, antibiotics in kind of third world countries where they're able to buy them kind of over the counter online and just use them. We are actually in a situation in the UK and where owners are able to do that with antivirals, and they're able to get hold of them and they're able to use them. So where are we now in terms of the 18 months of legal medications? There's a paper that we've recently submitted to help support information out there for vets that are wanting to use these medications. So we know that, as I said, 85% of these cats are surviving if we get the dose right for them. We know that we can still vaccinate these cats. We have a small number of cats that will relapse, usually within those first few months of coming off the medication. And we're suspicious in those that maybe we've just not achieved remission in those cases. We can certainly control the clinical signs, but maybe the underlying disease process isn't completely controlled. And I love the idea, this kind of, with the silly infection where it's just sitting there grumbling in the background, with the potential that if we've got an abnormal host response, we're going to be getting recrudescence of the disease process happening. We are seeing vets do fewer techniques, fewer tests are being applied. This does make the diagnosis less secure, more likely presumptive, but it does mean that we're getting to these cats often earlier and we're getting some really good survival times with it. What also does surprise me is that I work with a group of vet specialists, and we still have people in the UK vets in the UK that don't know about the availability of these products, that don't know how to get hold of these medications. So again, trying to disseminate that information has been a real struggle to get that information out there. Vets still struggling with diagnosis, still struggling with dosages. They're starting to reach out, which is really promising, but it is a real worry because if vets and the people they work with aren't able to convey to their owners a security in their knowledge about this disease process, these owners will then go elsewhere, will source the information from other people, and might not get an appropriate treatment or management process for it. Other concerns that I certainly have from a small animal clinician point of view, which is going to have an impact from a global kind of security point of view, is that with cost of living, it's going to be a price punch. That's going to be a problem when it comes down to getting hold of the medication. As we have more players coming onto the market supplying these drugs, I suspect, unfortunately, that the vets are going to be more inclined to mark up these medications as they would do any other medication out there. And actually, the margins that are going to be there because we have competitors within the situation are going to be lost to the client. I'm worried that we're going to get more drugs sourced illegally, and I'm also really worried that we're going to have limited diagnostics, so some of our security in our diagnosis is going to disappear, and potentially that's going to impact on our cats as well. We're also seeing people wanting to use antivirals to manage gastrointestinal infection. And again, the worry with that is that if we're selecting for systemic infection of a resistant viral strain, what's going to happen in the future if we get recrudescence of it, if there's another house fire and it comes out? So hopefully we've got just a couple of brief videos on here. So these are 3 cats that have FIP. The one on the left there was little Jellybean, unfortunately had hydrocephalus. And we have another one that's got effusive disease in the middle. And then we've got Zadie here, who kind of, as you can imagine, thought could hit the table and really fails on that one. Now, Jellybean, unfortunately, wasn't able to make it here today. Jellybean is in our FIP biobank because it came at a time when we didn't have access to these medications. And we've got over 200 cats, about half of which have FIP, that we're doing ongoing studies on. But now we do have medication, and unfortunately, you can't see the video here, I'm not too sure why. Zadie was the first cat I ever treated. I used the Vetclury on them. they started to improve within 48 hours. They went on to have a complete course and they're in remission. And that was in 2021. So we do have this ability to treat these cats, and we have that out there now. But I think what's going to be really interesting over the next day is to discuss about how we try and best manage the security of the use of these agents, and how we work towards getting a better pipeline of getting them validated, get the information out there to try and avoid a Wild West of situation where owners are able to access these drugs, whether or not they believe that the vet has got the diagnosis right. That's it.
Thank you very much, Emi. We have time for questions.
This is, sorry, just a fascinating topic. I saw you with the screenshot from FIP Warriors, and there's a ton of wild stuff in there. One of the things I think about a bunch, at least folks can't tell from the States, the regulations around companion animal health, we regulate products and tests, certainly, but the incentive to just improve companion animal health, to solve the companion animal disease without zoonotic concerns, without livestock concerns, kind of isn't there. And therefore the regulatory muscle with that is also not there. And then thinking about the owners, like you were saying, the individual owner is not incentivized to do much for the community. They want to treat their cat. I'm just wondering about the interaction between clinical vets and, I don't know if it's state or federal government or other agencies that actually get something like this moving forward. Like how do you get to this point even? Is that coming from government? Is that just people interested in the welfare of the animals?
I think part of what I think the main issue that we have in terms of the pipeline that's happening is that the world has changed. So it used to be really traditional in terms of getting medication. There was a very, very set process of being able to get it through the licensing process and getting it out there and then disseminating that information. And these days, as soon as the patent's out there and with the support of social media, it's very, very easy for these other players to come in and start producing these drugs and then ship them in small quantities. I've seen cases where the owners have bought what they believe to be antivirals online that have arrived to them labeled as vitamin C, and then has worked as an antiviral agent. I don't believe what they had in those capsules was vitamin C. So we know that the companies that produce them are very, very savvy in what they do, and they know how to get around the various legalities for that. I don't have a good way of getting around it. I understand that one of the reasons why it's been very slow to get these towards the market is the lack of desire, the lack of global market for the cats that they have in comparison to the livestock market, in comparison to the human market for antivirals. And I also understand the real concerns that the companies have for trying to bring a drug to the cat market, because bless them, they're pesky little creatures and they do some really odd things when we give them medications. They're really prone to getting kind of kidney issues, and if any of those things come out in the pre-testing, then that would put a real limit on the humans, the ability to bring it forward from a human marketing point of view. So I can really understand the reason to delay from a pharmaceutical point of view. And I believe one of the reasons why the GC376 has been slow to come to market, one of the reasons, has been because they've been wanting to test it for use in people for the management of COVID. And yes, it means that their focus is in that direction, which has diverted it away from cats. I tried looking earlier on it in terms of environmental impact about use of these antivirals and what will happen when they get into the environment and whether that's going to result in viral resistance. We know that use of some of these antivirals there has been detectable viral resistance occur. And that is also a real worry. What's going to happen, for example, if a cat is treated for enteric infection or a cat is treated for FIP and has enteric infection and then goes on to shed it into the environment and that becomes wild. We can see that these viruses can go around the world. You know, how we've ended up in a wildlife coronavirus in kind of Northwest Ireland in the cattle there. we can see that these odd transmission points can occur, and it can be very easy to get the resistance occurring. We can also see that spillover from animals to people. Yeah, I mean, we know at the moment kind of FIP doesn't transmit to people, but if things move forward in the future, if we start getting more alpha coronaviruses affecting people, how is that also going to impact on it? What we don't want to do is for it to just disappear out of our control.
Thank you. I think we'll have to finish there, although I'm sure there'll be many questions to follow. Can I now pass on to my co-chair, Lucy Davison, who will introduce our next speaker. Thank you.
Thank you very much. We're now moving to the part of the companion animal session where we're going to hear short abstract presentations from a series of speakers We've tried to timetable it to continue within the same themes. So our first speaker is Dr. Marsha Wallace, who will be talking about host susceptibility in FIP.
So I am a clinical research fellow at the Royal Veterinary College and the University of Oxford in Lucy Davison's lab. And what our lab has been focused on the past several years is actually pioneering whole genome sequencing for complex disease in dogs and cats. And so this is a stark contrast to the way that it's been handled in the field prior to this. So for dogs and cats, there have been these small SNP arrays. So looking at only a tiny amount of the genetic variation that's there. This is also quite different from a lot of the focus in disease in companion animals, which has been monogenic. And so this is, the first time that really we're looking and digging into the host susceptibility of these complex diseases like diabetes. And so then the pandemic hit, and we got some funding and switched our focus to actually applying these same kinds of approaches that we're using to look at these complex diseases. And looking at it specifically for coronaviruses in cats and dogs. And so we've had a couple of talks on FIP, and that's one of the things I'm going to be talking to you about today. I also have the canine respiratory coronavirus side of it. If you're interested in that, I have a poster on that. And so the heart of it for FIP really, though, is that this feline enteric coronavirus is ubiquitous. And most of these cats recover quickly, they recover fully. And then rarely, some of these cats end up developing months to years later this fatal FIP. And the question is, who are these cats? Why are they the ones developing it? What is really going on here? And so this is kind of the mystery behind FIP. And there have been a couple of genetic studies done looking at the potential host susceptibility to this, because that's the big question. Is it random, some of these cats that are getting FIP, or is there a host genetic component to this? And based on the predisposition of some breeds, specifically, for example, Burmans, to get FIP, that would suggest there is, in fact, some kind of host genetic susceptibility. So there are a couple of these studies that have been done. Again, these were on the small SNP array, so just looking at a tiny fraction of the genetic variation, and they were able to find a little bit, but we know that there's a lot more to the picture here. As a different approach, there's also been a look into the transcriptomic side of things. Or what are the genes, what are the pathways that are being changed in this disease? And so what our lab has done for the first time is we have kind of combined these kinds of angles, but at a whole genome kind of level, looked at the whole genome sequencing, as well as the transcriptomics, as well as combining some other data that I'm going to be talking to you about. And so this is kind of the heart of the study. And so we've got the whole genome sequencing that we've done. We add in the transcriptomics, and we add in a lot of other things as well that I'll talk a little bit more about in a couple of slides. And the heart of it here is that we're working with small cohorts. This is a high noise level environment. And so if you can only follow up a finite number of those genetic variants, you need a way to be able to prioritize, rank those variants, the most promising candidates that may be having a role for follow-up in larger cohorts. And so the work that I'm going to talk to you about today is our work with the whole genome sequencing, as well as phase 2 here. And so this has been adding in 105 cats, 10,000 variants, and we've got domestic short hair as well as a variety of other breeds. And what is currently underway as well is following up the most promising set to date in an additional 200 cats. We've been able to make this bigger and better as we've gone along in time by additional funding that we've gotten. And we've actually gotten funding from UKICN, as well as a collaboration with APHA and their funding through DEFRA. So on the transcriptomic side of things, this has been led by Tom Hiron in the lab. And so essentially looking at different tissues, liver, lung, mesenteric lymph node, what was very clearly coming out were these differentially expressed genes in interferon response pathways. And so this is quite interesting in terms of its parallel to severe COVID-19 patients and human patients. So we take that transcriptomic data, we take the whole genome data, and one of the things that we've also done is we've added in a lot of resources, a lot of annotations, a lot of data from other places. And so this includes things like impact prediction on the genetic variants. How is this predicted to impact the protein? We've also pulled in human GWAS. So what are the genes that are associated with a variety of different autoimmune diseases, for example? We've also got the UK Biobank data, which has been really useful, and that starts to tie in as well that we've brought in a lot of the results from the human COVID-19 consortia. And then lastly, what we also include are the big cat databases, big cat database. Feline 99 lives. And so this allows us to have an allele frequency comparison, because again, we're working with these small cohorts. And so what we want to do is we want to compare it to Feline 99 lives, which allows us an extra 350, say, whole genomes for comparison of allele frequencies. This gives us a much better comparison for being able to follow up those variants that truly are different, say, in the FIP cohort of cats. All right, so we've got whole genome coverage here at 30x. The initial whole genome panel was with 12 domestic short hairs with FIP, 12 controls, and then an additional 22 samples that we had available in-house from other projects. And I think this really kind of highlights what I mean by before, with just the SNP arrays, we're only looking at a tiny fraction of the available genetic information. It was 60,000 variants. And what we've got here with the whole genome here is 40 million variants. And so for the first time, we can actually see what genetic variation is even present. And so as I mentioned, we put in all of that other information and data together so that we can do this evidence scoring kind of a framework so that we can rank these variants, prioritize these variants for follow-up in these additional cohorts. So what you see on the left here is after the follow-up with our phase 2 cats. And so this is a Manhattan plot showing that we have I should also mention that these are adjusted p-values. And so you can see that we have some really nice hits here showing up on this Manhattan plot with a few examples listed on the right. And so these are genotype stacked bar plots. And so we've got the FIP cases. We've got of the full cohort, domestic short hairs, as well as all of the other breeds. Just the domestic short hairs alone that have FIPs. And then in that first and that third column, the control groups. And so you can see how the genotype profiles for some of these genetic variants changes between the cases and the controls. Oh, and I should also mention that with these particular examples, it's really interesting to see, again, what we're looking here is that host genetics. So these variants are residing in genes that are involved in antiviral activity or some type of immune-related process. And so when you look at the broad number of these kinds of hits, and you look at them in terms of odds ratio, what I have here on the left-hand side is side by side, each one of these rows is a different genetic variant, and side by side with what functional evidence is available for each one of those variants. So as I mentioned, these variants are residing in genes, some of which are in fact previously shown to be associated with human COVID-19 severity or found in the UK Biobank cohort. And many of them are involved in a variety of different immune-related evidences, say, for example, human GWAS autoimmune disease or just Go term, so immune processes just the gene's function. One of the things I also wanted to note, though, is that you'll also see some of these variants where there is no functional evidence listed there, and so these are quite novel findings. And so that also shows that, you know, the stats, if they're strong enough to stand on their own, we're still following up all of these targets. I should mention that all of these variants are targets that we're following up. And so, yeah, it'll be quite interesting to see which of these novel kinds of associations end up panning out through our phase 3. So in summary, we've got this RNA-seq data where we've shown differentially expressed genes that are involved in antivirus and interferon responses. We've got this whole genome sequencing evidence scoring approach that's able to identify variants associated with FIP that are in genes that are involved in immune responses, including interferon-stimulated genes. And some of these genes are known to impact COVID-19 severity in humans. So together with this RNA-seq work and the whole genome work, we can see that there is this critical role for the host immune response and type 1 interferons in FIP pathogenesis. And so as I mentioned, we've got phase 3 underway in more cats. I also wanted to mention too that our lab is doing a variety of kind of related and ongoing work. And so this includes actually looking at genetic variants, so host susceptibility in cats that are SARS-CoV-2 positive. Also, we're looking at the feline MHC, actually digging into the viral sequences themselves of cats that have FIP. And also we've got some funding from PetPlan to look at the functional impact of some of these in the immune system. And so we will be looking for a postdoc, as I'll mention in a slide or so. And so I think it's also important too to keep in mind the kinds of translatable goals and objectives and the kinds of things that we're accomplishing with this kind of work. And so what we've got, you know, are these novel genetic factors for susceptibility to severe coronavirus-related diseases. We've got the potential here for novel treatments, better understanding of how the animals are responding to coronaviruses, as well as there have been some hints in the previous talks, preparedness for some of these delayed kinds of phenotypes that we might see with COVID-19 patients, or, you know, the preparedness for future pandemics. I would like to acknowledge the entire mascot team, especially Sarah for extracting the DNA, Tom for doing his work on the transcriptomics, and we've got Emi and Judy for their access to these samples. I'd also like to thank all of our collaborators at APHA and our other authors as well. There's a picture that's not showing up here. It is our lab at, oh, that is not right, sorry. It's our lab in front of a giant rocket ship. So, yeah, so join us. If you're interested looking for a postdoc, come join us.
Thank you very much, Marsha. Do we have any questions?
Thanks for a lovely talk. I was wondering if any of the variants in your genome sequencing were either in like the regulatory regions or the transcription factors for the differentially expressed genes you identified by transcriptomics.
I'm sorry, what was your question about the transcriptomics?
So you found some of the genes were differentially expressed or increased.
Right.
I wonder if to link that back to your genomics data, was it in either the regulatory regions of these genes or the transcription factors that activate expression of these genes, potentially?
So, in short, yes. We've kind of got examples from the full spread, and definitely a couple hundred of the targets that we're currently following up, we did see as being differentially expressed in the transcriptomics. And when we're looking at these variants, we are, of course, looking at the 5 kb flanking region, so that includes enhancer regions as well.
Thanks a lot.
Thank you for this very nice talk. I just have 3 questions if I can remember them now. But one, in terms of your negative cohort, how do you know that you have a true resistant phenotype? And then my second question is, concerning your SNPs, are you looking at non-synonymous versus synonymous SNPs, or are you just looking for association with any of your SNPs? What I'm asking here, are you looking for causative mutations? And then I'll stop right here.
Yes, so we're definitely looking for causative variants here. And so in terms of, are we looking at synonymous and non-synonymous? Yes, we're looking at everything, seeing what associates. And then one of the things that you might have seen on one of the slides is one of those variants was in fact, as an example, a missense variant. And so that's where the impact of that variant comes into play in the evidence scoring framework. And so something that is, say, a missense variant then has a higher score than, say, a comparable synonymous variant. And your first question, sorry, was what again?
How do you know that they're...
There's a PCR test.
So Emi might be able to answer that.
Oh.
So the negative cohort that we use, certainly the ones that came out of the FIP biobank, they're all cats that were euthanized for reasons other than FIP, that have had histological evidence or clinical evidence of another disease process. They're not directly comparable in terms of a lot of those cats are going to be significantly older, given they were dying of heart disease, diabetes, cancer. But there was no histological evidence of FIP or that disease process happening. We know that about a quarter of those cats had detectable coronavirus within their system, typically at low levels, and we have attempted to do sequencing of that virus. It's never possible to have a perfect control group, but this is as much as we can do from that point of view.
Maybe last question. Do you think you'll be looking at disease tolerance or disease resistance?
That's a good question. Yeah, that's a good question. I guess the only way I can answer that really is we'll see what ends up panning out and what those hits are in terms of the mechanism that seems to be most likely in play. Sorry.
Okay, thank you very much, Marsha. Okay, same species, subtle shift of virus. Sorry, use the microphone. Same species, subtle shift of virus now. We're going to hear from Grace Tyson from the University of Glasgow about her work looking at the seroprevalence of SARS-CoV-2 in domestic cats. Thank you.
Hello. So I'm Grace. I'm a PhD student at the University of Glasgow, and I study infection of domestic cats with a little virus called SARS-CoV-2. You might have heard of it. So a bit of background, first of all, about SARS-CoV-2 in cats. There's actually quite a few species that are known to be susceptible to SARS-CoV-2, mostly through infection from infected humans. This isn't just domestic cats in the felid group, it's also lions, tigers, fishing cats, a whole host of different felids. And this has obvious implications for animal health. We've seen how much it has affected the human population, so we're worried about animals as well. And there has also been interspecies transmission reported in cats. It's not just all from humans. Under experimental circumstances, we've seen cat-to-cat transmission, but we've also seen mink-to-mink transmission in the wild and transmission amongst wild white-tailed deer in the US. And this has kind of led to a bit of concern about animals, particularly cats, becoming reservoirs for the virus and potentially passing it back to humans. We know of a case in Thailand in which a cat infected their vet. So it is possible that felids may have a role in the ongoing pandemic and further evolution of SARS-CoV-2. A bit about immunity to SARS-CoV-2 in cats. So we know that cats can mount quite a robust antibody response against both the nucleocapsid and spike proteins, and this can resist reinfection from further viral challenge. This has been demonstrated experimentally. And we can use this immune response to measure it via serology. We can use ELISA to detect the presence of antibodies, but also pseudotype neutralization assays, which not only show us how strong the antibody response is, but we can also look at the response to different variants. So we've looked at the response to 8 different variants so far. We published a paper at the beginning of this year in which we looked at the original ancestral strain along with Alpha, Delta and Omicron. And we've now had a look at more closely at Omicron, but also at BA2, BA5, which are sort of subsets of Omicron, as well as BQ1.1 and XBB. So a bit about our previous findings. We published a paper in Emerging Infectious Diseases which used samples from April of 2020 through to February of 2022, so over a year ago now. And in this, we just did a neutralization assay and found an overall seroprevalence of 3.2% of cats had neutralizing antibodies to SARS-CoV-2, at least one variant. And when we sorted these samples by which variant they produced the highest neutralization titer against, we could see that when we detected the variant in cats, it was quite soon after it emerged into the human population. As you can see, Alpha was first detected here in the human population, and it was detected in cats here. And this indicates that there was probably repeated cross-species jumps from owners into their pet cats, rather than the virus mutating and evolving independently in the feline population. And it's likely that the sort of what we can see here of these extinct variants, such as B1, which is the original strain, which is long extinct by the start of 2022, these were still being detected as the highest titer in cats. It's likely that this is because of immunity that is still being detected from previous infections. So what were our objectives for this new study? We wanted to find an update. This was over a year ago, the previous study. So we wanted to find what is the current seroprevalence in UK cats. Has it gone up or has it gone down? We also wanted to find out, are the newer variants such as BA2, BA5 that have emerged into the human population in the UK still emerging into the feline population? We wanted to find out also how strong are the titers against Omicron and post-Omicron variants. Is there still that existing immunity that we've seen to the older, more extinct variants? And we also wanted to do a bit of demographic analysis to look at risk factors. So a bit about our samples. Our samples we obtained from the University of Glasgow's Veterinary Diagnostic Service. These were from pet cats or cats from shelters generally. They were basically samples that had been sent for diagnostic testing by vets, and whatever was left over, we were allowed to test. And we had 4,385 samples, so a decent sample size. They were slightly skewed towards male samples, and they were predominantly from England, despite us being located in Scotland. There wasn't too many from Wales or Northern Ireland or elsewhere, unfortunately. The majority of our samples were from 2021 and 2022. Most of our samples were domestic or moggy cats. But we did have 33% pedigree, which is actually far more than the population. We think that in the UK, around 10% of the feline population are pedigree or purebred cats, so they were a bit overrepresented here. We also had quite a young cohort, with a median age of 3 years and a mean age of 5 years. So a bit more about the methods we used. First of all, we generated pseudotype viruses. We used a HIV core and embedded SARS-CoV-2 spikes from whatever variants we wanted to test on these. And these were useful because pseudotype viruses can only go through one cycle of replication, so it means that we can test these in containment level 2. We then screened all of our samples at a fixed dilution, basically to determine the presence of a neutralizing immune response. We then titrated these out using serial dilution, any of the positives, to determine the strength of the immune response, and also tested it against different pseudotype variants to see what were the levels of cross-neutralization we could find. And finally, we used an ELISA, which detects non-neutralizing antibody as well. So the useful thing about this is that it can determine if a sample that is positive on our neutralization assay is indeed positive, or whether there's something like bacterial contamination in our samples which is killing the cells in our assay rather than it being actual antibodies. So from these tests, we can categorize our samples either as positive neutralizing, so positive on both the ELISA and the neutralization assay, positive non-neutralizing, so positive on the ELISA but negative on a neutralization assay, or simply negative. So how this neutralization assay works, I'll go over it quickly. We make our pseudotypes using a 3 plasmid system. We transfect our cell line with the SARS-CoV-2 spike of the variant that we want to test, the HIV GAG-POL gene and a luciferase gene to produce our pseudotypes. Then, when we use them in the actual assay, if we have serum which has no neutralizing antibody present, we can incubate with the virus. Nothing will happen, so the pseudotype viruses should be able to infect our cell line, which are HEC293s, which express ACE2, which is the receptor for SARS-CoV-2. Therefore, luciferase will be produced as we transfected with this firefly luciferase gene. And so when a substrate is added, we'll get bioluminescence. And conversely, if antibody is present, this will prevent viral cell entry and no bioluminescence will be produced. So we get a nice inverted correlation between the amount of bioluminescence produced and the neutralizing antibody concentration. And we define our titer as the dilution of serum which results in a 90% reduction in bioluminescence. So what was our seroprevalence this time around? There was actually a rise in overall seroprevalence. We had 3.2% were positive neutralizing in our previous study, and it has gone up to 4.4%. But overall, when we include the ELISA results, it's up at 13.9%, so a lot higher. We can see that the neutralizing seroprevalence peaked in autumn of 2022, down here in the dark bar, and the overall seroprevalence actually peaked quite recently. The neutralizing seroprevalence has been pretty stable along here since autumn of 2021, and this is likely down to longer lived immunity from early infections combined with new infections. When we break this down by what variant our samples had the strongest titer against, we can see up here that the mean titer per season in our feline samples seems to kind of mirror. This is data from GISAID, which is for the proportion of human sequences that they received that were of different variants. We can see that we're kind of getting a little bit of mirroring of these waves, if you like, in our feline samples. However, along here, as you can see, things get a little bit more complicated. And this is because BA1, or Omicron, and its descendant variants appear to be a bit less immunogenic in cats. And the current peaks in feline immunity, they're nowhere near as defined as human waves, and things are getting a lot more complex now we've had so many viruses moving into the feline population. So we calculated the mean overall titer against each variant, regardless of what variant the sample had its highest titer against or when the sample was taken. And you can see that Omicron is significantly less immunogenic overall. This gives a picture of cross-neutralization as well. It's far less cross-neutralized too. We can see that when the highest titer was against B1, so the original ancestral strain, you get a bit of cross-neutralization of alpha and delta, but far less of Omicron and subsequent variants. Again, when the highest titer is against alpha, you get some cross-neutralization of B1 and delta, but very little against Omicron and its subsequent variants. When the highest titer is against delta, you get a little bit of cross-neutralization of Omicron and BA2, but still low. And then when we look at Omicron itself, when the highest titer of a sample is against Omicron, there's pretty much no cross-neutralization. One sample had a bit against Delta, but it's just not a very immunogenic variant in cats. So when we break this down by our demographic groups, if we look at breed, there's no significant difference between the average titer of domestic and pedigree cats, so the strength of the immune response is similar. However, when we look at the difference in actual seroprevalence, we can find that when the ELISA results are included, pedigree cats are significantly more likely to be infected than domestic cats. However, this isn't the case with neutralizing immunity. And when we look at specific breeds, Bengals and Siamese cats appear to have slightly higher seroprevalence rates than other cats. This isn't significant overall, but when we look at the highest seroprevalence, Bengal, against the lowest, which was Maine Coon, there is a significant difference. So there appears to be a trend towards pedigree cats being more seroprevalent. When we look at location, we actually had quite an uneven distribution of where our samples came from. So there was no analysis by postcode area. However, we could do a bit of analysis with Scotland, England, and Welsh samples. There was no significant difference in the amount of seroprevalence in these 3 countries. When we break this down by age, there was no significant difference between the mean titers of positive and negative samples. So what we did was we split our samples into 5 different age groups to do some further analysis. And when we did that, we saw that cats aged between 2 and 5 years were significantly more seropositive, and that's both neutralizing seropositive and when we include the ELISA results as well. However, this is not down to there being a stronger immune response in these younger cats because there's no significant difference in titers between the age group. It's more likely to be down to husbandry or behavioural factors in this sort of teenaged group. And finally, when we look at sex, there's no significant difference in titer between the sexes. However, when we look at seroprevalence, when the ELISA results are included, male cats are significantly more positive than female cats. However, this is not the case for neutralization. So again, there is a trend towards higher seroprevalence in males. So a little bit of a caveat. There was slight sampling bias in our study. Basically, because of where we got the samples from, the majority of cats will have been in veterinary care in some way, either under observation or the samples have been sent in for diagnosis of an infection. So yeah, our sample set was disproportionately ill. Also, our pedigree cats were overrepresented, as I said earlier. And third, we had no sequence data. This is all serology work. So the highest titers could be used to infer what might be the infectious variant, but this might be inaccurate. We can only use serology to infer this. Some further work that we're hoping to do is to look at the seroprevalence in dogs. We know that dogs are also susceptible. They're also in close contact with humans and indeed cats. So we want to look at what's the current seroprevalence in dogs and characterise that immune response. We also want to look at immunity to the nucleocapsid. There appears to be a lot more cross-neutralization of feline coronavirus with the nucleocapsid because that's more conserved. The spike protein changes a lot between discrete viruses. And we want to continue to screen against newer and future variants of SARS-CoV-2. So finally, just some conclusions. There was an overall seroprevalence of 4.4% neutralizing and 13.9% overall. And through serology, we can still see that mirroring of human waves of infection in the feline population. Omicron appears to be less immunogenic than other variants, and pedigree cats appear to have a higher rate of seropositivity than moggies. There's also a higher seroprevalence in male cats, and while age doesn't affect the strength of the immune response, cats aged 2 to 5 seem to have significantly higher rates of seroprevalence. So from these results, we believe that SARS-CoV-2 still remains quite an underappreciated problem in UK pet cats. We still don't know, population-wise, what the seroprevalence is, and there's no real availability of testing for vets. So that is really important to kind of look towards in the future. So that's me done. I just want to say thank you to all my co-authors. My Twitter's here, and also Sarah, who did a lot of work. She's over there if you want to ask her any questions. She did a lot of work on this study too, so I want to say a special thank you to her as well. And I'll take any questions you might have. Yeah.
Thank you very much, Grace. I think we do have time for a couple of questions. I've just noticed actually on the online timetable we've got a missing 15 minutes. So actually it works in our favour, so we're okay. We have nothing scheduled between 14:45 and 3 o'clock, so we'll just bring the 3 o'clock abstract forward. So it does mean we have got some time for some questions. I noticed one very enthusiastic one. Oh, there first, okay.
I've got a whole bunch of questions, but I see there are others, so I'll stick to one. I also feel compelled to comment. The transition of the slides, super slick, was great. I've taken a bunch from this already. The question I have is about time. I think time is such a complicated thing in this factor, especially you saw some really long-lived serologic responses. I think the mirroring of the the variant waves was interesting, but I wonder how you can account for, the proportions will change, but at least in the States, I assume here as well, just the sheer number of cases across the variants was really pretty different, and therefore the amount of cats exposed to those variants, did you see, not proportional, but just like count-wise, those same kind of things?
When we look at the sort of dominant variants, and when we look at the variants to which each sample has the highest titer against, we can only infer from that, okay, this is likely to be the infectious variant, but we don't know. Like I said, we don't have serum samples, so I don't feel confident enough in saying, yes, this definitely was, for example, a Delta infected cat, and we can definitely say that there were this number of cases in this month. I think you would need to do sequencing, PCR for that. We've tried to do PCR. It is, when you're doing it on a population-wide level, when you're not actually targeting cats that are displaying symptoms, it's very difficult to find positives, very difficult.
I'm laughing because I can empathize. I'm literally thinking on the same thing, yeah.
Yeah, so I just wouldn't feel confident enough saying this definitely was infected by XYZ to answer your question, really. Yeah.
Really nice talk. Just a couple of questions. How infectious, as in productive infection, is SARS-CoV-2 in cats? Does it release a lot of virus, or is it getting in but not getting out, or what? How does it work out?
So I honestly don't know the answer to that. There will be people here who will know the answer to that better. I know Angela's lab's done a lot more to do with actual sort of infections in the lab. I just do serology, kind of looking at the virus on a population level. I don't have experience of any kind of infections in the lab to try and quantify that. So I'm honestly not sure.
And just in terms of the timing, so you're getting the samples when the cats are coming for treatment, is that right? A lot of them.
Yeah, so basically the VDS is a diagnostic lab, so vets from all over the country will send samples saying, we think this cat has FIP, or we think this cat has feline calicivirus, will you test for this? And then if the owner is consented, we will then test whatever's left over in the serum or the plasma or whatever we've been sent. So they're from cats that are either ill or under observation, and they want to find out a different diagnostic thing. We test the leftovers.
But is that illness due to SARS-CoV-2, or is it just coincidental?
Sorry?
Is that illness due to SARS-CoV-2, or is it just coincidental seropositivity in the cats at the time of?
So it's just coincidental. We know that. Basically, we know that the immune response in cats, or the humoral immune response anyway, is quite long-lasting. So we've seen that it does tend to wane over time. There have been a few cats where there have been multiple samples taken, and we've seen the titers go down but not disappear. So basically, the immune response can stick around for quite a while. So it is likely we're still seeing infections that happened perhaps even in 2020, and we're only just testing the cat now, and it has a titer because it was infected in 2020. That doesn't mean it's had a recent variant. That doesn't mean that it's a recent infection.
So cats are better equipped than humans to handle SARS-CoV-2, is that right?
Who knows? Who knows, quite possibly.
I think we'll take one more question. I think over here was first. Thank you. Sorry, making you run with me.
I've got the hobbles.
Do you have any data regarding the status of COVID-19 in the households where these pets resided?
So because these were collected kind of from these leftover samples, we weren't able to actually sort of feed back to the vet or contact them. This was just a separate study. This wasn't, they hadn't sent their samples to be tested for COVID. So let's say if 6 months down the line, suddenly the vet hears, oh, your cat had COVID as well. The owner's like, what? So we haven't really been in contact with the households as much. We don't know what variants, for example, the humans had, when the humans might have been infected. That would be incredibly useful to know, because we can kind of classify, okay, when and what time frame did this infection happen? Are they likely to have got it from a human, or are they do they have, say, did the human only have COVID at the very start of 2020, but the cat has a really strong Delta titer, so it's probably likely to have got it from somewhere else. That would be incredibly useful to look at, but unfortunately we don't have that data.
Thank you very much.
Thank you.
Thank you. Great to have a lot of discussion on that one. Our final speaker of this session before we break, we're sticking with the same virus this time, but we're moving species, but ferrets still count within our companion animal space. So our final speaker is Dr. Amanda Seekings, and she's going to be talking about SARS-CoV-2 in ferrets.
Thank you. So yes, I will be talking about SARS-CoV-2 in ferrets and using ferrets as a model for what we might see infection in ferrets, but also as a mild asymptomatic model for what we may see in humans. So many of you, everybody will be familiar with SARS-CoV-2, and many will be unfortunate to have experienced SARS-CoV-2 infection. Some of us even more unfortunate to have experienced it more than once. So this study is looking at longevity of infection and immunity, and also what happens when reinfection occurs after several months. Looking particularly sort of at this phase. So we see here Delta infection, and 5 months later we see potential reinfection with Omicron. And looking at that scenario, looking at humoral immunity and the ability for reinfection and onward transmission, and also looking at virus evolution. It is important to understand this in the context of animals, in the context of potential animal reservoirs. So we have studied this in ferrets using a model that we previously established at the start of the pandemic. So the project was entitled SARS-CoV-2 Longevity and Reinfection Dynamics with Virus Evolution, looking at primary infection and looking at immunological parameters, and also touching on a little bit about fomites and bioaerosols as well, but also focusing more on the potential for homologous variant and heterologous variant reinfection and transmission to naive contacts. The third part of the project is looking at virus evolution in the context of host immunological pressure. That part of the project is still in progress, so I'll be focusing on the first two for this talk. So just an outline of the experimental design. So we used female ferrets, 12 to 23 weeks of age, and in the first phase we housed them in 4 groups of 4. 2 groups received the Delta variant, and 2 groups received Omicron BA.1, and we did a direct intranasal inoculation. And so we collected nasal wash and swab samples for the beginning of the infection, and we looked to see for viral clearance. So as I said at the beginning, ferrets don't become sick with SARS-CoV-2 infection, so we were able to keep the animals on, and we tested serum samples over a course of 5 months before we initiated the reinfection. So in this scenario here, we took all 16 from the first phase, and we divided them up into separate groups. So here we see a block, but each block of these is replicated 4 times. So in each group, we have one of the directly infected ferrets from the first phase, and we introduce direct contacts that have not previously been reinfected. And we have the 4 different scenarios. So we have previously infected with Delta, reinfected with Delta, previously infected with Omicron, reinfected with Omicron, and underneath we have the heterologous reinfection, so previous infection with Delta, reinfection with Omicron, and the other way around here, Omicron and then reinfection with Delta. So just showing the results here from the Delta variant infections. So for logistical purposes, for when we got to the second phase, we had to do the initial infection in 2 separate groups, 2 different times. And so what we see here are the nasal wash, oral swab, rectal swab, and fur swab data, and each individual ferret is represented by a different line here. And what we can see is oral nasal shedding at the beginning of infection, and then viral clearance from about 10 days onwards. And what was nice to see here is that what we saw in group 1 was largely replicated in group 2. So we were fairly confident that they were okay. And then when we did the same experiment in Omicron, we saw there was oral nasal shedding at the beginning. It didn't last as long. It was a shorter duration at lower levels, and compared to Delta at lower levels. But what was a little bit unfortunate was actually what we saw in group 1 was not quite the same as what we saw in group 2. So group 2, there was only 2 ferrets that were really shedding, not the other 2, so slightly different. But keep that in mind for when I show you the results of the serology and the reinfection in phase 2. So just to summarize, with Delta, we saw infection of all 8 ferrets, and shedding was predominantly oronasal, and similar in both groups. And this graph here shows the periodic time points over the 5 months prior to reinfection, and looking at the antibodies detected by virus neutralization tests, to show that this is the point of reinfection, or just prior to reinfection, that there was a high antibody titers. When we look at Omicron infection, we can see we saw that group 1 and group 2 did not shed as much comparatively, lower compared to Delta, and seroconversion was only really observed in 4 of 8 ferrets, so one of the groups, and very low titer in the second group. And yes, those circles were just showing the titers just prior to going into reinfection. So here we're showing the previous infection with Delta and reinfection with Delta, and we see here that there is 1 ferret shedding, so this is a directly infected ferret. I'm not sure how clear it is here, but the solid lines are the ones that were directly infected, and the dotted lines or dashed lines are the contacts, and all the way through for the next graphs, they are color coordinated. So if you've got a solid green line of the direct infected, it was in contact with these 2 dashed lines that are the same color. So you can see which contact belonged to which direct infected. So in this infection, 1 ferret was shedding, but there was no transmission to any contacts. And when we look at the neutralizing antibodies and ELISA data, so this is the point of reinfection, and then we took serum samples at 14 and 28 days post-reinfection, and we have up here the directly infected and very low levels of the contacts. So 4 out of the 8 contacts were weakly positive by virus neutralization, but negative by ELISA. And over here we've got the same data just plotted with the ELISA on there as well. And one thing that was interesting to note was actually this purple ferret here. So prior to reinfection, this ferret had the lowest neutralizing antibody titer. And actually, if I go back 1, that was the 1 ferret, the purple ferret, that was the one that was shedding. So these are the graphs from previously infected Delta and reinfected with Omicron. And you can quite clearly see here there's no shedding, no transmission. A little blip here, but not very convincing. Now, when we look at the serology results, we can see high antibodies by neutralizing antibody and ELISA results from the direct infected, but the contacts had limited neutralizing antibodies against Delta or Omicron and negative ELISA results. And in this graph here, we did so where we had the heterologous reinfection, we did the virus neutralization with both variants separately to see if there was a difference. Now going on to the previously infected Omicron and reinfected with Omicron, and we can see that there was one ferret that was shedding at a very low level, and limited transmission here to one ferret at low level. When we look at the serology results, we can see generally a lower level compared to when we saw Delta infections, but all direct reinfected had neutralizing antibodies, but only one out of these were positive by the ELISA there. One of the contacts had low neutralizing antibodies, but this was also negative on ELISA. Now a slightly different picture with the ferrets that were previously infected with Omicron and reinfected with Delta. All 4 of the Omicron directly infected were shedding, oronasal, and there was transmission to one contact, can be seen here. And this, so if you remember, this was the group that previously infected with Omicron didn't really have a high antibody response, wasn't really robustly infected. And so what actually we see here upon reinfection with Delta is something very comparable to when we infected naive ferrets with Delta. And when you look at the serology results here, you could see an increase in neutralizing antibody titers after reinfection with Delta, and the direct reinfected positive neutralizing antibodies and ELISA, but the contacts had lower neutralizing antibody titers, and only one was positive by ELISA, and that one was the one that was shedding. In this study, we also wanted to look at environmental samples, and we looked at looking at dissemination of virus into the environment in the cage, in the feed bowl surfaces, looking at the water bottle, and these were all negative by PCR from the Delta and Omicron groups. We previously looked at air samples in the cages and in the room outside the cages. We previously did a study where we were able to detect viral RNA from ferrets detected infected with Delta. And so here we wanted to see whether we could see the same with Omicron infection, and it was not the case. And there were some weak positives at the very early on of the previous first infection, not very convincing though, not very much, very low level. And so this could be a potential characteristic difference of Omicron infection versus Delta. So some more work is still in progress here. We do want to look at virus evolution to see whether there's any mutations that have acquired coming out of the ferrets, and whether the reinfection has introduced additional mutations. We've also got pending the histopathology and immunohistochemistry from the tissues collected from the ferrets at the end of the study, although previous tissue collections in ferrets have not necessarily resulted in any immunohistochemical detections, but it will be interesting to see what the immunopathology might look like. And there is also cellular immunity, cell-mediated immunity that we would want to explore. So just to conclude this work that we've looked at so far, the upper respiratory and gastrointestinal shedding was observed from all directly inoculated naive ferrets. There was higher shedding from Delta-infected ferrets compared to Omicron, and that the neutralizing antibodies could be sustained for up to 5 months in Delta-infected ferrets, when this actually did provide protective immunity against reinfection. with the Omicron variant. Was quite interesting to see that actually the neutralizing antibody titer of greater than 32 was required to protect against reinfection. So it was quite nice to see that difference. And so this data really contributes to understanding longevity of SARS-CoV-2 immunity and reinfection. And we would like to continue this work and see how it fits into the context of re-exposure and the risk of animal reservoirs and the emergence of novel SARS-CoV-2 variants. So just a final slide to acknowledge everybody involved in the project. It's quite a big effort from the APHA virology, animal sciences and pathology team to perform such a large in vivo experiment for such a long duration. And also the funders for the project, and Wendy Barkley for providing the Delta variant. And one final note, we have a vacancy at the moment, so if anybody is interested in joining us, please speak to me, please speak to Sharon, but just to note that the closing date for applications is Monday, so be quick, thank you.
Thank you very much indeed. That's a great study, a lot of work. Are there any questions? Yes.
What a marathon. Potentially silly question, but nonetheless I think it's important. With your Omicron, did you check your stocks to make sure that it was still infectious or there was no loss of titer before you infected, or after you infected primarily, to make sure that there wasn't a stock issue rather than a susceptibility issue?
Yes, we did. We did back titrate our inoculum. So yeah, we don't know. It could just be, we infected them with as much the highest titer we could grow, which is also comparable to sort of the minimum infectious dose, really. So I think ferrets really do need quite a high dose to be able to actually show that infection, and it could be that Omicron's just, not as good as Delta or other.
Hi, thanks for the talk. Just in relation to the environmental sampling, I was just wondering, what were the environmental conditions when you did your sampling? So temperature, ventilation in the room, for example, and I wondered, did you consider testing other environmental conditions, like, for example, decreasing the temperature to simulate, I don't know, something like a meat processing plant where there's a suggestion that at lower temperatures the virus will persist better on surfaces or in the air? Just wonder if you expand on that a bit.
Yeah, that's really good. So we didn't alter the temperature or alter anything. We just took these as opportunistic samples that we could while we were running the experiment. So it was run at room temperature, and there's it's in BSL-3, so we've got our standard air changes and negative pressure going on in there. So
I suppose do you think it represents a room that we would normally, or animals would normally be in, or in terms of ventilation, let's say? I'm guessing a CL3 would.
Yeah, potentially not exactly mirroring it, but it was more whether we could detect anything in that scenario, and Omicron didn't seem to be as detectable as you might.
Thank you.
So it's about 18 degrees with about 20 air changes an hour. So your living room with the window open, maybe?
So maybe a naive question, but might have relation to the last one too. So given how closely related ferrets are to mink, the sort of relatively low transmission from one to the other, how does that relate to the mink farms where it appears to be very high? Is it again the sort of poor conditions, or is it the sort of poorer welfare and the stress of the mink, or different variants of the SARS? I mean, how do the two relate?
Many things, I think, is the answer. Probably also density as well. I think, here in this, we've got one directly infected and 2 contacts, and you probably need many more to replicate that.
The space in the cages is actually quite generous legally for an experimental model in comparison to animals that are in fur farms, etc.
Just a slight comment. This is work on mink. There was a paper from way early on talking about the the similarity is in the ACE2 receptors, but they're really different tissue distribution across mink and ferrets, upper respiratory and lower respiratory, which might explain illness and therefore transmission.
Thanks for a really good talk. I just got a quick question. So the antibody levels between the first and the second infection seem to stay quite high without any waning. Was this expected, or was that a bit of a surprise?
To be honest, we did not know what to expect. I mean, most of the studies that we've seen in ferrets didn't monitor for so long, so we didn't know. We had expected it to drop, but it was quite nice to see that they were still maintained at the high level. Thanks.
Really excellent study. I was just curious about your ELISA, since the results typically are kind of the opposite. Usually you see higher ELISA titers. Were you using a commercial kit?
Yes, we were, commercial, multi-species ELISA.
Okay, so
yeah.
Do you have any idea if that's why maybe you had lower ELISA titers than neutralization titers? I would have expected the opposite.
It's possible, yeah. We don't know. I mean, it could just be also the difference in the immune response that the ferrets are experiencing as well. So that could affect.
Thank you. Sorry. Thank you very much. Thank you very much to all our speakers in this first part of Companion Animals. I'll hand you back over to Sharon.
Okay, thanks. I'm going to take the opportunity, we've got some other slides that we'll put up and circulate during the coffee break, but yet another plug for one of our sponsors. So the Microbiology Society have sponsored John, who's going to talk shortly, and they've also sponsored the early career scientists, so for oral presentations and poster presentations at this meeting. And I would also just like to let you know that they are doing a special collection of coronavirus publications in the Journal for General Virology and the Journal for Medical Microbiology. So these journals are run by the Microbiology Society, which is a non-for-profit organisation. So if you are thinking of publishing any of your material and you might like to put them there, then they will be highlighted in the special collection. Thank you. We're breaking for coffee now. Just out here, we've got a little bit longer, so please take the time to have a look at the posters. Thanks very much.